So today I have been asked to present our talk on the structural biology of the coronavirus spike protein. So to start off, I'm going to go, I hope everyone is awake at this point but I'm going to share, it's going to be a short talk. So what do actually structural biologists do? Structural biology is a multidisciplinary field. It intersects biology, biochemistry, and biophysics, and they usually use complex and expensive techniques such as NMR, X-ray crystallography to understand the structure and dynamics of the biomolecules, such as proteins, RNA, DNA, etc. And through understanding those structures, they try to focus on the function of these biomolecules in our cell and body. So so as you can see in this schematic diagram of the coronavirus, the spike protein, it forms a trimer. So three proteins coming together and protruding outside the surface of the coronavirus. And each of these protein is about 1,273 amino acids long. So you can think of amino acids as building blocks of the protein. So if protein is thought of a building, then amino acids would be the bricks of that building. And they are about 180 to 200 kilodalton in size, which is in rough estimate nanometer range in length. So the technique they use to resolve the structure of coronavirus spike protein is cryogenic electron microscopy, or cryo-EM. I'm showing here the one of the figure of one of the instrument. I hope everyone is seeing on the right side. It's a huge instrument and very expensive instrument. And the way this technique works, that it flash freezes the sample by using liquid nitrogen usually. So you can think of as if, like, if you're taking a picture of a running person, the picture would come out as blurry. So you would have to make sure that a person is standing still so that it can take a clear picture, right? So the idea is same here, because in the solution, the proteins tumble, moves around. So it's a dynamic situation. So you want to freeze them and stand still so that you can take that picture. Now, can you take a camera and take that picture? Absolutely not, because in our visible spectrum, that's the micrometer range. And as I said, the proteins are at nanometer range. So you have to bombard these proteins with electrons, which wavelength is a nanometer, and that gets deflected off, and you see them. And there is a complex software that analyzes those images and reconstruct the structure of the protein. So that's how they solve the structures of the protein. On the left side, you can see the closed conformation of the protein. And on the right side, you see the open conformation of this protein. And each of them has three different colors indicating three proteins coming together. And on the closed conformation, the RBD, receptor binding domain, is in a closed conformation. And on the right side, when it is coming to the host cell and binding to the ACE2 angiotensin converting enzyme 2, it kind of opens up. And that's how it initiates the interaction with the host cell. And in this schematic diagram, you can see that interaction. So once they establish that interaction, then it fuses with the cell membrane, as you can see in this schematic diagram, and eventually it initiates that infection at host cell. So from this understanding of the structure of the spike protein, it allowed the scientists to come up with the idea with developing a vaccine targeting against this receptor binding domain, so that would stop the virus to initiate any interaction with the host cell and eventually prevent that infection. So in summary, it was important to understand that the structural biology was key to develop this vaccine against the coronavirus. And it's not just one case. Structural biology has been playing a key role in laying the foundation for numerous therapeutic targets against diseases such as cancer, and it would continue do so in the future as well. I'm going to end my presentation with the verse of the Holy Quran in chapter 67, verse 4. Allah Ta'ala says, "Who has created seven heavens in harmony? No incongruity canst thou see in the creation of the gracious God. Then look again. Seest thou any flaw? So in this beautiful verse, Allah Ta'ala is asking us to ponder over his creations. And if we look into galaxies, planets, and stars, we see those mega structures, and we see how harmonious and coherence in his creations. And even at the molecular level, at the atomic level, when you are going into minor details of it, you see the similar level of coherence. And that's the beauty of Allah's creation. And that's where Allah Ta'ala wants us to ponder over his creation, and we should be able to so that understand the Holy Quran. So may Allah Ta'ala enable us to do so. Ameen. Jazakallah.